everyone, and welcome to Miss Estruch Biology and this entire Topic 7 theory video. If you want help to make notes as you go in a structured way, then download my free workbook that matches this video. If you're looking to boost your grade even more though, through things like live tuition lessons with me in a private group and with other teachers and examiners, plus having your work marked by me and other examiners, exclusive discounts on my resources for free, plus so many other benefits, then you want to be joining the Biology Study Club. This is my monthly membership where you get loads of lessons per week for less than the price of one tuition lesson. So click the link in the description to see if you're interested in joining to boost your grade now. AQA Topic 7, and it starts with 3.71, Inheritance for A-level only. There are lots of key terms in this topic that you could be asked for a 1 or 2 mark question. So I recommend that you screenshot this or when the answers are there, or turn this into flashcards because these are essential to learn. Genotype, that's the genetic constitution of an organism, which basically means the alleles it has for a particular gene. Phenotype is often 2 marks because it's the expression of the genetic constitution for 1 mark. Second mark is interaction with the environment. Homozygous and heterozygous I haven't seen come up as definition questions, but it's important that you know what those mean. Homozygous meaning you've got 2 alleles that are the same for a particular gene. Heterozygous is 2 different alleles for the same gene. Recessive allele and dominant allele have both been assessed before. Recessive is an allele that's only expressed if there is not a dominant allele present, whereas a dominant allele will always be expressed in the phenotype. Codominant is when both alleles for a gene are equally dominant and are both expressed in the phenotype. Multiple alleles is if there are more than 2 alleles for a single gene. Sex linkage is when the gene of interest is located on the X chromosome. So locus means the location of the gene on a chromosome. Autosomal linkage is when genes are located on the same chromosome. So it's going to be 2 different genes are found on the same chromosome, but it's not the sex chromosomes. Epistasis is when one gene modifies or masks the expression of a different gene at a different locus. Monohybrid and dihybrid don't come up as definition questions, or at least they haven't, but it's just knowing what they mean. So monohybrid, mono meaning 1, it's looking at the inheritance of just 1 gene. A dihybrid cross would be looking at the inheritance of 2 genes. So inheritance, then we're going to go through all of the types that you need to know for AQA, but there's different ways to present what allele you have for a particular gene, and that's what I mean here by this coding. This is what we'll use when we do Punnett squares or flow diagrams to work out predictions of what you might see in offspring. So for a simple monohybrid cross, that's what you would have done at GCSE. There's just 1 gene with 2 alleles, the dominant or recessive. So you pick 1 letter to represent the gene, capital is dominant, lowercase is recessive. But then when we get to codominance and multiple alleles, we need to do the coding differently. Because for codominance, it means both of the alleles are dominant. So we can't pick 1 letter and have it capital for dominant because they're both dominant. So instead, you pick a base letter to represent the gene. And then superscript, you have the 2 alleles. So a dominant B, dominant A. And it's not going to be the same letter because they have to be capitals to show their dominance. Multiple alleles, because it's more than 2 alleles for a gene, again, you can't pick 1 letter because that only gives you 2 versions. So we use the base letter to represent the gene. Superscript, you pick the allele letter. And for all of these, if they haven't told you what letters to use, you can pick any letters that you want. But I recommend that you pick letters where the lowercase and capital letter are completely different shapes, like all the examples I've done here, so that it couldn't be confused by an examiner whether you did put the capital or lowercase. Sex linkage, you have the base letter as the chromosome, so whether it's the X or the Y chromosome, and the allele is superscript next to that. There won't ever be anything written next to the Y chromosome because you only get the gene on the X chromosome. Autosomal linkage, this is where we said you have two different genes on the same chromosome, and usually this is just done with simple monohybrid inheritance, so one letter to represent the gene and then capital and lowercase for dominant and recessive, and that's for both genes. Same with epistasis. This is when one gene masks or modifies the expression of the second gene. So we've got two different letters representing those two different genes, and then dominant and recessive is shown by capital or non-capital letter. So let's have a look at some examples, starting with monohybrid inheritance. Cystic fibrosis is caused by a recessive allele. If two carriers reproduce, what's the probability they'll have a child with cystic fibrosis? And on top of that, what's the probability they will have a girl with cystic fibrosis? Now you can pause, have a go. I'm going to go straight through. One thing to be really aware on these questions is they're doing this more often now, where they specify the sex of the child as well, when it isn't a sex-linked inheritance question. So what that means is we need to do our Punnett square to work out the probability of having a child with cystic fibrosis. And it's always 50% probability of having a girl, 50% probability of having a boy. So whatever your answer is for the cystic fibrosis, you then half that because that's 50%. So here are our two carriers. I've picked the letter F here because the capital and lowercase are different shapes, but you could pick A. That's even more obvious if you had a capital A, lowercase a. So we can see here that on our Punnett square, if those are our two parents' genotypes, I've then put in the possible gametes that each parent could produce. I've then done our cross to show F with this F, the two capitals, lowercase and capital, lowercase and lowercase, and capital and lowercase. And because cystic fibrosis is caused by a recessive allele, they will only have cystic fibrosis if they are homozygous recessive, because there's no dominant alleles there in their genotype. So we have 25% probability that these two parents will have a child with cystic fibrosis. Then we need to also account for a girl with cystic fibrosis. We need to half that. So times that by 50% or half it is 12.5%. Next, we have a codominant example. Cows can be red, white, or roan in colour. Red and white are both dominant. If two roan cows reproduce, what's the probability they will have offspring that are red? Again, pause now and you can have a go at this Punnett square. I'm going to go straight through it. So for this one, parental genotypes, red and white or roan. Red and white are both dominant. So that means those are our two dominant alleles. Now, they'll probably give you more information than that in a question to make it explicitly clear that roan isn't a third allele. They'd need to either tell you it was codominant or give you a hint towards that a bit more. So here, red and white are both dominant. And when red and white are the codominant genotype, you get roan. This is roan here, is when you've got both the red and the white being expressed in the phenotype. And yes, I would personally say that's more brown than red, but it's called red for some reason. So there's our two parents who are both heterozygous. They have that codominant genotype. So if we do our Punnett square, both parents would have one of the white alleles and one of the red alleles. We then combine those in our Punnett square, and we can see we'd have roan, white, roan, and this would be our only one that would be red. So 25% probability of having a red cow. Now, when you do these Punnett squares or flow diagram, it's really important that you are showing the parental genotype if that's not already been given. Then the gametes, and the difference between the two is the gametes are separating those two alleles, whereas the genotype, they're both written next to each other. You then would have to show the offspring genotype and the matching or corresponding phenotype for that genotype. That's why I always leave a space and write the phenotype in the box so it's matched, and then we've got our probability. So then we've got a blood group example, and this is not on the spec. It's just a common example because it models codominance and multiple alleles. So blood groups, you could be blood group A, B, AB or O. And this is referring to the type of antigen you have on the outside of your red blood cells. So some people have antigen A, some have antigen B, some have both antigen A and B, and some have no antigens at all. So that's what the blood groups are corresponding to. And the genotypes for this are, to be blood group A, you could either have 2 copies of the dominant A allele, or 1 copy of the dominant A and 1 copy of O, because O, blood group O, that recessive allele, well, it is a recessive allele. Blood group B, so you could have 2 copies of dominant B or B and the recessive O. Because O is recessive, you are only going to be blood group O if you have 2 recessive O alleles. And then blood group AB is the co-dominant genotype, because if you have A and B, they're both dominant, they are both expressed in the phenotype. So we've got co-dominance and multiple alleles because we have 3 alleles. So if we have a look at a genetic cross example, as I said, this is not on the spec that you need to know blood groups. So they would have to give you information about it to be able to work it out. But I went through it because it's a relatively common example, or at least it used to be. So we're told we've got parents with blood group AB and blood group O reproducing together. In the question, we're then told IA and IB are co-dominant and IO is recessive. What is the probability that they will have an offspring with blood group A? So the first step is working out the parents' genotypes. And if they are AB, they must be A and B, that co-dominant. And if it's O, that is recessive, so homozygous recessive for O. Then we can put in the parents' gametes. Then we can do the cross, and then we can add in the blood group, so the phenotype corresponding to those genotypes of the offspring. And we were asked the probability of having an offspring with blood group A, and that is 50%. Then we've got a sex linkage example. So colour blindness, again, you don't need to know this as an example. In fact, all of the examples I've done are application. There is no genetic condition or disease that you need to know for AQA. You'd always be given the information and then you do the genetic cross. So colour blindness is caused by a recessive allele, or at least one type of colour blindness is caused by a recessive allele found only on the X chromosome. If a non-colourblind male reproduces with a female carrier of the allele, what is the probability their children will be colourblind? So step 1 is always working out the genotypes of the parents. And when it's sex-linked, you get marks for including the sex chromosomes in the genotype as well. So you do have to include that. For the male, it's XY. For the female, it's XX. And remember, you only find the allele on the X chromosome. So the female here is a carrier, and we're told it's recessive. So there'd be X. I've picked the letter R, so it's really clear, the dominant versus recessive. So X, capital R, lowercase r. And that's a times, that's not part of the genotype. That is crossed with the male, which is X, capital R, Y. Because they are not colourblind, they only have the dominant allele. So then if we have a look at our cross, you might have noticed it's actually slightly changed. So I just went and edited it to really emphasise a key point, which I'll get to. So we have the male gametes here, X, capital R, and then the other gamete would just be the Y. Then we've got the X, lowercase r, and X, capital R for the female, the mother. Now, I've actually done that the other way round, how I've written it here, because there it was the capital first, but it doesn't matter. Your answer ends up the same. So we then fuse together the gametes to show all of the possible genotypes in the offspring. And then I've added the corresponding phenotypes for those genotypes. And you have to state the sex as well as the characteristic linked to the gene of interest. So here it's female, normal vision. They may actually call that colour vision. In the older exam questions, they used to say normal, but they've started to not use that now because it implies if you don't have colour vision, then it's not normal. So they would say colour vision or normal vision. But it's female and they have colour vision. Female colour vision, male colour vision, male colour blind. So you have to state the sex and also the characteristic in the phenotype. And for this one, that's 25% probability of having a colour blind child. Epistasis then, that was when one gene influences the expression of another. And this is often seen in colours. So it could be coat colour in mice. It could be coat colour in Labradors. It could be fruit colour of vegetables. So these are the common examples that you see. And we're going to go through an example and then do the cross. So let's have a look at coat colour in Labradors. The first gene controls whether pigment is going to be produced. And if you have the dominant version of that gene, so dominant allele, then that codes for pigment will be made. The recessive version codes for no pigment. So if a dog is homozygous recessive for this gene, they will not produce any pigment, and that's how you get the yellow dog colour. So that is no pigment has been produced in the fur. Gene 2 controls which colour pigment is expressed if they are able to make pigment. Capital B is black fur, lowercase b is brown fur. So that's how we get these three different colour furs, or hairs if it's dogs, from two genes. So let's have a look at the cross here. We are told straight away the parents' genotypes. And you can pause now and have a go at, first we're working out, what would the phenotype be of these two dogs? So we've got capital E, lowercase e. This was the one that controls where the pigment is made. This is the gene that controls what colour that pigment is. And for this one, they'd both be black because they've got the capital E, which means pigment can be made. Capital B means the pigment would be black. So if we then did a genetic cross, first of all, we have to work out all of the possible gametes both dogs could make. And because this is now a dihybrid cross, meaning the inheritance of two genes simultaneously we're looking at, you have to show all the possible combinations that you could get. So you need to show just one copy of each gene, so one letter E, one letter B, but all of the possible combinations. So we could have the capital E with the capital B, the capital E with the lowercase b, the lowercase e with the capital B, the lowercase e with the lowercase b. So we get 4 possible gametes for both of those parents. So that means our Punnett square is going to be 4 by 4. It's much bigger. And we now need to show all of the possible genotypes. And I've then added in all of the possible phenotypes as well. So here we go. We can see all of those genotypes and the corresponding phenotype. And we have 9 black, 4 yellow, 3 brown. So we've got a 9:4:3 ratio for this. So that actually goes into dihybrid crosses. A dihybrid cross is the inheritance of 2 genes at the same time. And we've just done one example, but here is a more straightforward example where we don't have epistasis involved. So we've got a round and yellow pea, a wrinkled and green pea. So one gene is coding for shape, one gene is coding for colour. And with all genetic crosses, we need to have the parent's phenotype, the parent's genotype, the possible gametes the parents could make, the offspring genotype, the offspring phenotype, and then the proportion of each phenotype. Sometimes 1 and 2 might be given to you, sometimes they aren't. So for this one, the genotypes, you would need to have information to know this, but we've got round and yellow, and in this instance, both parents are homozygous for both genes. They would have needed to tell you that for this parent, you wouldn't know otherwise, because it's a dominant characteristic, round and yellow, so they could have either been homozygous dominant or heterozygous for both genes. So round and yellow, wrinkled and green are the 2 recessive characteristics. They had to be homozygous recessive for both genes. Possible gametes then, the round and yellow can only give dominant alleles. So 1 dominant R, 1 dominant Y in every gamete. And this one here can only give recessive alleles. So all of the gametes would have 1 recessive R, 1 recessive Y. So that means we could do a Punnett square, but actually every offspring will get this gamete and this one. So combining them together, that gives us offspring that are heterozygous for both genes. And that would mean they'd be round and yellow. So 100% are round and yellow in our first generation in this dihybrid cross with these parents. Now you could be asked to then cross F1, which means first generation. And that would now mean we're crossing 2 peas that are heterozygous for both genes. And this is now when we'd get 4 possible gametes for both of those genes. So capital R with a capital Y, capital R with the lowercase y, lowercase r with the capital Y, lowercase r with the lowercase y. So looking at all the possible combinations of getting just one of the R's and one of the Y's in every gamete. And this is then when you get that big Punnett square again, the 4x4 Punnett square. And if we combine all of those together and then add in all of the phenotypes to the corresponding genotype, we can see we get a 9:3 to 3:1 ratio. And in a dihybrid cross, if you have two parents that are both heterozygous for both genes, like we saw here, then you will always get 9:3:1 because you'll always have this pattern of gametes. Therefore, crossing it together, it doesn't matter what the genes are, you'll still get this outcome. So sometimes for 1 mark, you don't have to do the Punnett square and they just ask you and they tell you, you've got a dihybrid cross. Both parents are heterozygous for both genes. What will the ratio of the phenotypes be? And it's just 9:3 to 3:1 because it's learning that fact. However, there are exceptions. So if there is autosomal linkage, you won't get that ratio. If there's crossing over in meiosis, you won't get that ratio. And if there's epistasis, you don't get that ratio. So sometimes that is also asked as just a 2-mark question. Maybe it's to give 2 reasons why you might not get that, or suggest 2 reasons why you won't get that ratio. And it's those 3 reasons. And that leads us into what is autosomal linkage and how does it mean, or how does it result in you getting a different ratio? Now, I'm actually going to quickly go over crossing over in meiosis very briefly, because this actually is from topic 4, but you need to have an awareness of it for this. So crossing over of homologous chromosomes happens in meiosis 1, the first round of division in meiosis. It's when those homologous pairs, so the chromosomes, pair up at the equator to form a bivalent, meaning 2 chromosomes next to each other. The non-sister chromatids can cross over, get tangled, break and swap that section of the chromatid. And as a result, when you come to work out what gametes a parent might be able to produce, you get new combinations of alleles in the gametes. So that's the relevance here, because you might have thought, well, these parents could make these 2 alleles, but if crossing over happens, maybe they can actually make 4 different gametes. So autosomal linkage, this is when we have 2 different genes located on the same chromosome. And as a result, they have to be inherited together. So it's going to impact the gamete predictions, because I'm using the same example here, the 2 Ps, heterozygous for both genes, the capital R, lowercase r, capital Y, lowercase y. But now we're imagining that there's autosomal linkage. So we've got gene 2, which was for shape, gene 1 is for colour. They're still heterozygous for both genes, but we can see this chromosome has the dominant R and the dominant Y. This chromosome of the pair has the recessive R and the recessive Y. And that means after meiosis, you will have to have gametes that are capital R, capital Y, because this chromosome will be going into one gamete, this one will be going into the other. So we previously said when there was no autosomal linkage, there were 4 possible gametes that parent could make, but with autosomal linkage, there's actually only 2 possible gametes that these parents could make. So here was our original when there was no autosomal linkage, heterozygous for both genes, and here are all the possible gametes. So these 4 and these 4 for that parent. But now there is autosomal linkage, we can actually only create these 2 gametes. And that's the case with both parents because they both have this autosomal linkage. So that now means that this is the possible genotype and phenotype combinations we could get. So we have 3 yellow and round and 1 green and wrinkled, which is not our 9:3 to 3:1 ratio. So if there is autosomal linkage, that is how it's going to affect the ratio. But now let's add in though, crossing over as well. So here's what we've said so far. With these 2 parents, We would expect 9 to 3, 3 to 1 ratio, but if there's autosomal linkage, we actually had a 3 to 1 ratio. However, sometimes you're told that information and then you say, scientists actually observed the following results. And we can see we have 4 phenotypes, and here are the numbers they observed. They've got mainly yellow and round, then green and wrinkled, and then you don't have very many yellow, wrinkled or green round. And they would ask you, how is it possible they got these results? And the only way this is possible is if there is autosomal linkage and crossing over in meiosis, because that crossing over in meiosis created new combinations of alleles in the gametes, and that's why it was possible to get these 2 extra phenotypes. So let me show you a common answer for that. This is actually just modelling what we mean here. So here we had our autosomal linkage. We've got the recessive alleles for those 2 genes on this chromosome, the dominant on this chromosome. However, there was crossing over in meiosis 1. Those chromatids break off and swap. So as a result, we now have new combinations of alleles in the gametes. This was the original, so the non-recombinant, the 2 recessive alleles. This one at the end is also non-recombinant, meaning it's not being recombined and created through crossing over in meiosis. We've got capital R and capital Y. But these 2 options here, capital or dominant R and recessive Y, and this one is the dominant Y, recessive R, that is our new combinations of alleles in the gametes, and it's created these 2 new gametes. But crossing over is rare. So that means you won't get very many of these 2 gametes, and you'll get lots of this gamete, lots of this gamete. And that then leads into our answer. So explaining these results. Yellow and round, the top one, these are the 2 genotypes that you could get from the non-crossing over possible gametes. And you could also get the green and wrinkled from 2 lots of the lowercase r, lowercase y joining together. In fact, that's actually written here as well. So those are the most common, the top and the bottom are the most common because those gametes are not a result of crossing over in meiosis, and crossing over is rare. Now, the reason we have fewer green and wrinkled compared to yellow and round is there's 2 possible genotypes compared to only 1 for those phenotypes. And that's because green and wrinkled is recessive. So here we can see the heterozygous genotype still gives us yellow and round. So that's why there's the most yellow and round, then the green and wrinkled. The 2 phenotypes in the middle of the table are not as common, and that's because crossing over is rare. So we don't make very many of these 2 gametes. And that then means we don't get very many of these genotypes, which code for those phenotypes. So the key thing with a question like this is pointing out autosomal linkage occurred, if they haven't already told you, and also pointing out crossing over has occurred. But that's rare, which is why we don't get very many of these gametes and therefore those phenotypes. So sometimes the mark scheme does insist that you literally pick out which gametes were a result of crossing over, which were not, and link that to the numbers of each phenotype. So you could be asked to do a statistic on this, and it would be chi-squared, because you're looking at how many individuals fit into a category, or in this case for inheritance, how many individuals have a particular phenotype. And if we want to see, is there a difference between what we expect based on our Punnett square and what we actually observed, we can do a statistic to see if it's a significant difference between what we expected and what we observed. So here's an example. Going back to what we did at the start, cystic fibrosis caused by a recessive allele. We said we expected there to be 12.5% probability of it being a girl with cystic fibrosis. We could then actually look in the offspring and count what proportion did have cystic fibrosis and were a girl, and see what we expected. Is that significantly different to what we observed? Now, you wouldn't usually do that with humans because there's not going to be a large enough sample of the offspring from those 2 parents for it to work. But we've got an example here with corn, looking at the colour of the corn kernels. And in this example, we have got purple and yellow corn. Purple is dominant, yellow is recessive. Based on this Punnett square, we expect a 3:1 ratio of purple corn kernels to the yellow colour. So that's what we expect. We're then told that a student counted the actual number, so this is what they observed, and they got 21 purple kernels, 13 yellow. And we have to say, does that follow what we expected? So our null hypothesis would be there is no significant difference between the expected and observed frequency of the colour of corn kernels. And then we can fill in a table like this. Now, you wouldn't be expected to remember how to draw this table, but it has come up that they've given you this table with empty sections and you then have to fill it in. So you need to know that O is the observed, E is the expected. So here's what the student observed: 21 purple, 13 yellow, which means there were 34 in total. We said we expect a 3:1 ratio, but we need to turn that ratio into frequency. So that means we need to know what 75% of 21 is and 25% of 13. And I've calculated that here. O minus E is your observed frequency minus your expected frequency. So we've got that result here. Then we square that. So even if it was negative, because it's squared, it all becomes positive. And then we divide that by the expected frequency. The sum of this column is your chi-squared value. So we've got a chi-squared value of 3.176. You could be asked to work out the degrees of freedom, and that is the number of categories minus 1. We've got 2 categories, purple or yellow, so we have 1 degree of freedom. Now, if we were to look at this in a critical values table, it's a bit blurry, but we need to look at the P equals 0.05. That means that we'd have less than 5% probability that the difference between observed and expected is due to chance. We've got 1 degree of freedom. We've picked out the column that has 0.05. So we are comparing to this critical value, 3.841. And the critical value is like a threshold. You've got to see, does your chi-squared value meet or exceed that critical value threshold? And it has to meet or exceed it to be able to say there is a significant difference. So ours doesn't. Our chi-squared value is lower than the critical value. And there'd be a mark for stating that, which I've put here. So what that means is there is more than 5% probability that the difference is due to chance, not R, I should say is. And you do have to state difference and not just result to get the mark. So that means we can accept the null hypothesis, and there is no significant difference between what we observed and the ratio that we expected. Or in other words, the corn kernels did show a 3:1 ratio because it wasn't different to what we expected, and we expected 3:1. Next then, 3.7.2, populations. And this is where the Hardy-Weinberg principle comes in. It's normally assessed within the inheritance questions though as well. And Hardy-Weinberg is a mathematical model that can be used to predict allele frequencies within a population. So here are just some definitions to check. Turn them into flashcards if you want, because they are commonly assessed. Gene pool, that is all the alleles of all the genes within a population at one time. Population, this comes up as a 2-mark question now. All the individuals of one species in one area at one time. 2nd mark, that can interbreed to make fertile offspring. Allele frequency, the proportion of alleles within the gene pool. So here's the Hardy-Weinberg equation. And in fact, although the top one is the equation, you have to use the top and this one here simultaneously to get your answers. So you need to know that lowercase p, that is the frequency of a dominant allele in the population. Q is the frequency of the recessive allele. So a single letter by itself is representing the frequency of an allele. P is dominant, Q is recessive. And if you add all of those together, that equals 1, because that's all of the alleles for that gene in a population. P squared is the frequency of genotypes. Specifically, this one is the dominant genotype. 2pq is the heterozygous genotype frequency. Q squared is the frequency of the homozygous recessive genotype. And again, if you add all of the genotypes together, it equals 1 because those are all of the possible genotypes you can have for this particular gene. So in this question, cystic fibrosis is caused by a recessive allele. 0.02% of the UK population suffer from cystic fibrosis. What proportion of the UK are carriers? So step 1 is always identify from the two equations, what information have you actually been told? So we have been told that 0.02% suffer from cystic fibrosis. And cystic fibrosis is caused by a recessive allele. So to have cystic fibrosis, you have to be homozygous recessive. So 0.02% is Q squared. But we can't have it as a percentage, so I've converted it. Then we need to work out what we need to identify. And we've been told what proportion of the UK are carriers. Carriers, that would be heterozygous. So they want us to work out 2pq. And the strategy for Hardy-Weinberg is always work out which bit of the formula you have, then which bit you're trying to identify. And to do this, you then always need to work out what P and Q are. So Q, we know what Q squared is. So if we square root that, we then get what Q is. And now we've got Q, we can use this formula to work out P. Rearrange it, 1 minus 0.014. This is what P is. So now we are working out what proportion are heterozygous. So for heterozygous, it's 2pq. So we then just need to do 2 times P times Q. And that comes to 0.03. And they've just said proportion, so we can leave it as that, or you could write 3%, but do look carefully to see if they've asked for your answer as a percentage or just proportion. Next then, 3.7.3, evolution may lead to speciation. So variation, individuals within a population of a species may show a wide range of variation in the phenotype, and that is because of both genetic and environmental factors. If it's genetic variation, the primary source of genetic variation is mutations, but also genetic variation is introduced through meiosis and the random fertilization of gametes. Predation, disease, competition for the means of survival. Basically, this means there's going to be competition within populations. There's going to be disease, which means some individuals are more likely to survive, and there might be predators, and therefore if you're more likely to evade a predator, you're more likely to survive. And this then leads to natural selection. And the organisms with the phenotypes providing a selective advantage, meaning they're more likely to survive, they are also then more likely to reproduce, pass on those beneficial alleles to the next generation, and that's how we get this differential reproductive success, or in other words, the allele frequency for that gene might change in the gene pool. Now, all of this is in topic 4 as well as topic 7. And I go through that in more detail in the entire topic 4 video, because this is just a brief summary that they do in topic 7. And basically they do it so that you can be assessed on this content on paper 1 and paper 2 and paper 3. So types of selection. There are 3 different types. Stabilizing selection. So by type of selection, we mean the outcome of natural selection. Here's our original population looking at number of individuals for each trait. And the modal trait is the most common, or the middling feature, so medium color is the most common. The very pale, the very dark are not as common. And stabilizing selection does favour the average phenotype. And individuals with the extremes are less likely to survive, therefore less likely to reproduce, and therefore over many generations, you lose those extreme traits, and the middling trait that was already the most common becomes even more common. And the example that you learn in topic 4 for this is human birth weight. Directional selection also came up in topic 4. This is when one of the extremes provides selective advantage and is favoured in those environmental conditions. So maybe there's an environmental change, which now means this extreme trait provides the selective advantage. So over many generations, more individuals now have the extreme trait, the middling trait's less common, and the other extreme is almost completely lost. And the example that you learned in topic 4 was antibiotic resistance, because those with the most resistance were more likely to survive. The new one for year 13 is disruptive selection. And this is when both extremes are favoured, meaning they both provide a survival advantage. So if an individual has either of those extreme traits and allele, they're more likely to survive, reproduce and pass on that allele. And this can then lead to the process of speciation. Now, you don't have to know an exact example for this like you did for the other two, but an example could be maybe birds with very small or very large beaks are more able to survive because birds with very small beaks might be able to get their beaks into tiny holes in trees to eat insects, and the birds with bigger beaks are able to crack seeds and nuts and eat that. But if you've got a medium-sized beak, you're not very good at either, so you're not going to get as much food. So this then takes us into the concept of evolution, genetic drift and speciation. Evolution is a change in allele frequency within a population over many generations, and that can also lead to speciation. Speciation is the process that results in the creation of a new species, and this is going to occur when one original population of the same species become reproductively isolated. And this isolation means you now have two separate populations of the same species, but they do not breed to make fertile offspring at this point. And that's what reproductively isolated means. It means they cannot reproduce. Now, the reason that might happen is different, and we're going to come to that. But the outcome, if you've got these two separated populations that are no longer reproducing, both populations will have random mutations happening. But because they're no longer reproducing together, those mutations stay within this population. Mutations that anyone has in this population stay there as well. And that means over time, you get this accumulation of differences in the gene pools, so that if they were able to start reproducing together again, they'd be so genetically different that they cannot interbreed to make fertile offspring. And therefore they're now classed as a different species. So that is our process of speciation. And what causes them to be reproductively isolated is either a geographical barrier, which is known as allopatric speciation, or they might be in the same location, but they still don't reproduce. And that's known as sympatric. So allopatric speciation is when they're reproductively isolated because of some geographical barrier. And that could be a new mountain range, a body of water, but they just can't reach each other. All of the other marks would be exactly the same. So you always have that concept of they are reproductively isolated. You have to state whether that is geographical, allopatric, or sympatric, which we'll get to. That means that different mutations will accumulate in both populations, and therefore they'll go through natural selection and evolution for their environment. You get an accumulation of different mutations, and therefore they're now so genetically different they cannot interbreed to make fertile offspring. Sympatric, this is when populations become reproductively isolated, not because of a geographical barrier, but due to something else. And it could be their behaviour. So it could be that linked to the idea of courtship, or it could be as well that they are now producing gametes at different times of year. So they're fertile at different times of the year. So even if they are mating, they're not making offspring because one of the pair in that reproduction is not making a gamete. And then you get the same marks again. So for genetic drift, this is the change in the allele frequency within a population between generations. And there will always be genetic drift from one generation to the next, but continual, substantial change or genetic drift is when it's classed as evolution. And in smaller populations, genetic drift has a bigger impact because if you have a slight change in the number of alleles, in a small population, that's a big change in terms of the frequency, because if you just change 2 out of 10, that's 20% change. If you change 2 out of 100, that's only 2% change. So the smaller the population, the bigger the impact, and that's why smaller populations evolve more rapidly. Next then, 3.7.4, populations in ecosystems. And again, lots of key terms here. Population is a group of organisms of the same species living in the same area at the same time, and they can interbreed to make fertile offspring. Habitat is the part of an ecosystem in which a particular organism lives. Community is all the populations of different species in the same area at the same time. Ecosystem is a community and the non-living components of the environment. This bit here isn't part of the definition, but it's just being aware that ecosystems vary in size. Niche is an organism's role within an ecosystem, and that includes its position in the food web and its habitat. And this is going to be determined by adaptations to both the abiotic and biotic factors they're exposed to. Carrying capacity is the maximum population size an ecosystem can support. Abiotic factors, those are the non-living conditions of an ecosystem. Biotic factors is the impact of the interactions between organisms. So a bit more on these two, abiotic and biotic factors. We said that the population size and the carrying capacity are dependent on abiotic and biotic factors. Abiotic factors are the non-living factors, so that could be things like temperature, oxygen concentration, carbon dioxide concentration, light intensity, pH, soil conditions, and the key kinds of questions you get are either linking to something else in the specification to explain why any of those would affect plant survival or animal survival, and that might link to photosynthesis, might link to enzymes. And then it's knowing maybe links to how they could evolve. So that would be the idea of natural selection. If they're better adapted to that environment, they're more likely to survive, pass on those alleles to the next generation, and then eventually over many generations, much of that population will have adaptations to those abiotic factors. Biotic factors, that is looking at things like competition and predation. Competition can be either interspecific or intraspecific. Interspecific competition is when members of different species are competing. And the way to remember this, I think, is international travel, spelt inter, T-E-R, is when you're traveling to another country. So it's a different country. So interpacific competition is a different species. Intra is when members of the same species are competing. They'll compete for the same things. It's just if it's members of the same species, they're also competing for a mate. And this is for animals and plants. It's not just animals that compete. Plants would also be competing for water availability in the soil, carbon dioxide concentration, mineral ion concentration in the soil too. Predator-prey relationships, so this is still biotic factors. Looking at the interaction between the predators and prey in a food web, you get this typical shape graph, regardless of the species, where we've got the predator shown in blue and the prey shown in red. And you always see these three features. The size of the predator population is usually going to be lower than the size of the prey population. Both populations will fluctuate in size, and there's a bit of a lag time. You'll see a change in the prey before you see the change in the predator population. And this is because the predators are eating the prey. So there has to be more prey than there are predators to sustain them. They both fluctuate because as the predators are eating lots of prey, that causes their population size to decrease. That will then cause the predator population size to decrease. But because the predators have gone down, the prey can then increase, and this happens over and over. So the last little bit then is looking at sampling, so that you can look at population sizes. And the reason we sample is it's going to be more accurate and less time consuming than trying to count every single individual. But we have to make sure that the samples we take do accurately represent the population. So that would mean you have to do random sampling to avoid bias. And that's if it is a uniform area, meaning it's equal distribution. If it's not equally distributed, you do systematic sampling, which is a line transect or belt transect, to examine that change over distance. And then also it has to be a large number of samples to ensure your sample is big enough to accurately represent the population. Now, if you say large number, that should be fine. If you're going to state a number, I would say 30 plus, just to show it really is large. Now, the different ways that you can sample then. First thing is thinking about what type of organism you're estimating the population size of. So if it's slow moving or it doesn't move, like a plant, then you'd use a quadrat. If it's a motile, meaning moving organism, like a bird, you would use mark, release, recapture. If you've got uniform distribution, you do random sampling. If it's uneven distribution, it's systematic sampling, which is your belt transect or line transect. We're going to have a look at that first, and then we'll come back to the mark, release, recapture. So random sampling. First of all, you would lie 2 tape measures at right angles to create a gridded area. Then use a random number generator to generate your coordinates. That then tells you where to place your quadrats. You go along those 2 numbers of your coordinates. Depending on what the question says, will determine how you're estimating in your quadrats. Because it might say you need to do percentage cover, in which case you're estimating inside of your quadrat what percentage of it is covered by your plant. Or it might ask you to estimate the number, and in which case you do density, which means count the individual number of plants that there are there. Frequency doesn't tend to come up, but local frequency is the idea of you would use a gridded quadrat and just count how many of the squares it's present in. So it's slightly different to percentage cover, because that you're estimating the percentage covered. The local frequency is just, is it present or not in the tiny squares? Now you have to make sure it's representative. So you have to do this a large number of times, so at least 30 times, calculate a mean. And then if it's percentage cover, it ends there. If it's density though, meaning you counted the number of plants in the quadrats, you now have your mean number of plants in a quadrat, but we want to know it for the entire field. So there'd then be a mark for describing how you'd work that out. So you'd need to know what the area of the field is and the area of a quadrat to see how many quadrats would fit into that field. And then whatever your mean is, you're multiplying that by the number of quadrats that fit into the field. Another thing that they've become really picky on is if they've said describe how to work out the mean number, you also have to actually describe how to calculate the mean and not just say calculate it. So you'd need to say add up all of your 30 repeats and divide by 30. A line transect, this is used to estimate population size when there are that uneven distribution of species. So that could be things like on a rocky shore, if you've got a river in the middle of a field, and you'd place a tape measure to act as your line transect, going through all of those different sections so that you then do get information for all of the different parts. And a belt transect means that you've got your tape measure and you're lining up quadrats next to it. An interrupted belt transect is what is most commonly used. This means you place your quadrat at intervals along your tape measure. And it might be every 5 meters, every 10 meters. However far you start to notice a difference, that's the frequency that you'd need to place them to make sure you don't miss any important information. You still have to repeat multiple times though. So here would be your method if you were asked to describe a line transect. Method, that is. So place your tape measure at a right angle to the shoreline. So that would mean it's going through all of those different sections that we want to see. Place your quadrat every 5 meters. It doesn't have to be 5, but it's at stated intervals. Collect your data, just like we said last time, so either density or percentage cover. And repeat again a large number of times, calculate a mean. But for this one, the repeats would be multiple repeats at parallel, so in parallel to your original one, along the beach or whatever area it is that you're measuring. So then we've got mark, release, recapture for if you are trying to estimate the population size of a motile organism. Here is our method. So you need to capture an initial sample. You need to mark them. So that might be putting a bit of paint on a snail shell, for example. Then you release them back into the wild. You then leave them for a period of time after they've been released so that they can randomly disperse throughout the habitat. Then you take a second sample, and the total number captured in the second sample and the number recaptured, meaning the ones that have the mark on them, are recorded. Then we can use the formula to calculate our estimate. So to work this out, we'd need to do the number of organisms initially caught in that first sample, times by the number of organisms in the second sample, divided by the ones that had the mark on them. So that would then give us, in this example, we've got 20 butterflies were initially caught. In the second sample, there were 22 butterflies, 9 of which had the mark. So we do 20 times 22 divided by 9. That comes to 48.89, but we need to have whole numbers here because it's numbers of organisms. So I'm going to round up to 49 butterflies. There's always a mark for stating this formula when you have a method question for mark, release, recapture. So if in doubt, always state that formula. There's questions linked to the ethics of this sampling as well. So you need to consider that when you're marking the initial sample, that the way you mark them is not with a toxic chemical. So if you are putting on paint, it has to be non-toxic. It mustn't increase their chance of being seen by predators and therefore eaten and killed. And it mustn't reduce their chances of reproduction. So those are the ethics. Now, a non-ethical consideration of the mark is it needs to be weather resistant, because you don't want it to be something that's going to wash off when it rains, because then they're not going to be marked anymore. There's also assumptions linked to this. The estimate might not be accurate because the population size isn't actually constant, and we are assuming it is. And for a population to be constant, that means you have no births, no deaths, no migration. And that doesn't happen in any population. So we're assuming the population size is constant. In reality, it's not. So that's one reason why our estimate will be inaccurate. And also, we're assuming that the animals, when we release them and leave them to evenly distribute, we're assuming that they are going to evenly distribute. But in reality, they don't often do that, because lots of animals will probably huddle together near a water source or a food source, or where they feel safest. Then we go on to succession, and this is the change in ecological community over time. A primary succession starts with a pioneer species, and that is going to colonise. It might be bare rock or sand. You don't have any soil yet. You don't have to know any examples, but just to give you a bit of information, pioneer species could be things like lichen. Lichen are a mixture of fungi and algae, and they can survive in really harsh abiotic factors. And through the death and decomposition of the lichen, or whatever the pioneer species is, that is going to change the environmental factors, making them less harsh, less hostile, and you get this thin layer of soil, and then larger plants are able to survive there. And that might be things like mosses or smaller plants. And they will further increase the depth of the soil and the nutrient content, because they'll have roots that will start to hold the soil together. And as they die and decompose, they're going to add more nutrients to the soil. And this pattern continues over and over. Each new species is going to improve the environment so it's less hostile. But that then does mean that bigger species, plant species, can arrive and they'll outcompete the previous one. So the changes that organisms produce in the abiotic environment can result in a less hostile environment and therefore increases the biodiversity. And the final stage of a succession is known as the climax community, and this is dominated by trees. Now, to give you the marking points, because that was more a full explanation, the actual marking points on a long answer question are very brief. There's always a mark for saying it's initially colonised by a pioneer species. Next, it's saying the pioneer species changed the environment. The environment becomes less hostile for new species. There's an increase in biodiversity, and it finishes with a climax community. That would get you 4 marks on a 5 marker for describing succession. So screenshot that or turn it into a flashcard to learn those key points. So here's our summary from Succession. The species richness increases, the number of organisms increases. That's why the biodiversity is higher. So as succession occurs, we get larger plants. That is going to attract animals as well, and you end up with these more complex food webs. Now, you can prevent succession from progressing, and that is a technique to conserve habitats. And this is because as a succession moves through its stages, you ultimately get a climax community where you have lots of large trees. And those large trees will outcompete the smaller plants because they're going to be shaded. So if you just let succession occur, all of the area would be forest. And that's what it used to be in England, for example. It used to just be forest everywhere in the UK, not just in England. But then we cut down the trees, started to build. But you can manage forest as a way to maintain maybe one of the earlier stages of succession, to have one section of the forest thriving with grasses, another section of the forest thriving with smaller plants, and then some of the sections thriving with trees. And the idea here is you get a greater variety of habitats. That then means you've got a greater variety of food sources, and a greater range of animal species will be able to survive there, and also plant species. So you might do things like copacine or pollardine, where you cut down some of the trees, but you don't take out the roots, because then over time the trees can still regrow, so it's not going to be harmful for the environment. And also it means you get timber for furniture, or you could use the wood for fuel as well. So that takes us to the end of topic 7. Hopefully you found it helpful. And if you do want even more help, then you can scan here to get my A-level notes, or scan here to get my flashcards. But that's it for this video, and I'll see you in a video very soon.